Hi guys, today we are going to be doing our draw along. We are drawing protein synthesis. So I'm going to be basing some of my information off of this picture. And if you have colored pencils or anything like that, that's great. You don't need them. What we do need is to call this protein synthesis. All right. And we need to remember that there's two parts of protein synthesis: transcription and translation. All right, now I recorded this instead of doing it with you live, so that way you can pause wherever you need to pause if I'm going too fast. And you can also watch it over again if you want to kind of get ready for the test. All right, so this is the cell membrane. And let me make the nucleus a different color. This is the nucleus. And if this is the nucleus, that means that all the rest of the stuff in here is the cytoplasm. Is this on the screen? Yeah, that's the cytoplasm. All right, and we know that there's other organelles in here, but the only one it's not really an organelle. We're going to be thinking about the ribosome. All right, so what do we know is living inside of the nucleus, trapped inside the nucleus? We know that it's the DNA. All right, and if we zoom in on this guy here and unzip it, we might see an A, T, G, C, T, A. Let's make it 6. All right, T, A, C, G, G, C. Okay, so that's our DNA. We know that it cannot fit outside of the nucleus. It can't fit. So what has to happen? This DNA has to be transcribed. It has to be transcribed or copied onto a strand of messenger RNA. So the messenger RNA is going to come along. All right, and I'm coloring outside of the lines a little bit. And anywhere, it's just going to copy itself off of a half of the strand. So T is going to turn into an A. C is going to turn into a G. A is going to turn into a U. This is important. Remember, uracil replaces thiamine in RNA. A is going to turn into a U. G is going to turn into a C, and C is going to pair with G. I shouldn't say turn into, I should say pair with. So what we've created here is a strand of messenger RNA, and this can leave the nucleus. So it's going to exit, let me just draw a little. It's going to leave through one of the nuclear pores, and it's going to make its way out, and it's going to travel to the ribosome, and it threads itself into the ribosome. So let's just A, G, U, U, C, G. It came and it threaded itself into the ribosome like that. Every 3 nucleotides on the messenger RNA is called a codon. 3 nucleotides is called a codon. So here's one codon and here's another codon. The next player here is the transfer RNA. And when I make a transfer RNA, I kind of make it look like a T. Okay, when I make my transfer RNA, I make it look like a T. And what every transfer RNA carries with it is an amino acid. So what is going to occur here is the ribosome is going to be sending a message out to the cytoplasm, and it's going to be looking for transfer RNA. And each transfer RNA has, it potentially has 20 different amino acids. So once the transfer RNA figures out where it's supposed to go, it's going to bring its anticodon over to the messenger RNA. So the A is going to look for a U because we are in RNA. The G is going to look for a C, and the U is going to look for an A. Okay? A pairs with U, G pairs with C, U pairs with A. This is going to come and park itself right there, and it's going to leave the amino acid behind. It's going to leave it here. On this side, U is going to pair with an A, C with a G, G with a C. And well, these are called codons because they're on the messenger RNA. These are called anticodons. Anticodon, because it's on the transfer RNA. So these are going to come, drop off their amino acid, and what occurs next is the amino acids are going to link together. So this amino acid and this amino acid will be left behind, and they're going to link together with a peptide bond. Okay? Once we have a full chain of amino acids, what do we call that? That's now a protein. Okay? So some things to remember. Amino acids are the monomers of proteins. Okay, so the one Lego piece of the protein castle. And that's it. That's protein synthesis, guys. That's transcription. This is translation. Transcription occurs in the nucleus when the message that's carried by the DNA copies itself onto the messenger RNA. Once the messenger RNA leaves the nucleus, it comes out into the cytoplasm. And when it's here in the cytoplasm, it's going to be translated by the transfer RNA. Codon pairs with anticodon. We still follow the rules for base pairing. Okay? And as these transfer RNA drop off their information, they leave their amino acid behind. The amino acids link together in a chain, and that chain is a protein. So this picture looks exactly like this picture, if you ask me. All right. At this time, I want you guys to submit on Schoology. Okay, so go ahead and submit this on Schoology so that way I can give you some credit for your beautiful, beautiful artwork. Thank you.